In this video, we are going to continue our discussion of cytokine signaling, focusing on a new family of cytokines known as the interferons. And I'd like to start our discussion of interferons with a bit of a historical perspective. Interferons were actually the first cytokines discovered, although we now call them class 2 cytokines. You may remember in our last video we learned about class 1 cytokines, or hematopoietins. Interferons are class 2 cytokines, although in my experience, they are rarely referred to as such. You really mostly hear them referred to as interferons. But as you can see, they were the first cytokines discovered actually in the 1950s, and they get their name, I think, from that historical era, sort of the golden age of physics, when protons and neutrons and electrons and so on were being discovered and described. Very early and very simple classic biological experiments, virological experiments, revealed that when you infected cells in culture with viruses, as you can see here, you could get those viruses to grow in a dish if you gave them a type of cell or a type of tissue that they were able to infect. However, really early observations showed that if you did sort of sequential infection experiments where you infected first with one virus, you saw that it could replicate really well, but then if you infected with a second virus, its replication was relatively poor. And these types of experiments and similar ones where, for example, if you inactivated a virus and then you treated the cells with that inactivated virus, but then you went in with the live virus and infected, you would still see this kind of interference effect where previous exposure to viral material seemed to protect cells from subsequent viral infection. This led to the discovery that cells make some sort of secreted factor, some protein, as it was later discovered, that, again, interferes with viral infections. And so they named this particle interferon. And they did not know very much about what interferons were or even what cytokines were, but this was some of the very earliest work that helped us understand how the innate immune system operates. And we now know that interferons are, in fact, secreted proteins that are produced in very high abundance in response to viral infection. And they are specialized, I think, in many ways for interfering, again, with the ability of viruses to infect cells, to actually enter cells, to replicate their genomes inside of cells. There's many things that interferons induce in the cell that we'll talk about in this video. But even within this family, there are sub-designations of interferons, and we can see that there are sort of three subfamilies which are referred to by types. So there are type 1 interferons, type 2 interferons, and type 3 interferons. And here I want to clarify that interferons are class 2 cytokines, but within this class 2 family known as interferons, there are three types, 1, 2, and 3. And within these types, there are sort of prototypical cytokine ligands that we associate with the types. So type 1 interferon, primarily we're talking about the ligands interferon alpha and interferon beta, although there are others, but these others are more esoteric and much more rare. I think for an introductory video to interferons, alpha and beta is sufficient because they are the ones that are produced by far in the greatest abundance and the most frequently. And the type 1 interferons are also notable because their expression is ubiquitous throughout the body. Again, they represent a very primordial form of innate defense against viruses. And because most cell types are susceptible to one virus or another, it's a very conserved sort of feature of innate immune signaling that most cell types can make type 1 interferon and use it in response to viral infection. However, the other types are a bit more specialized. Type 2 interferon is primarily associated with the ligand interferon gamma, and this mostly is expressed by T cells, cells of the adaptive immune system. You can find examples of interferon gamma being expressed by other cell types, but I think it, again, by far the most common and the most abundant source of interferon gamma tends to be activated T cells. And so for that reason, type 2 is a bit unique among the interferons because it is more associated with adaptive rather than innate immunity. Finally, there are type 3 interferons, and these were actually only discovered in the early 2000s, and the main cytokine ligand that we associate with type 3 interferon is known as interferon lambda. And interferon lambda has a lot of overlap with alpha and beta. Type 3 and type 1 interferons are very similar. The main specialization of type 3 interferons is that they tend to be expressed at barrier tissues. You can remember from previous discussions, I've described barrier tissues as sort of portals of viral or pathogen entry into the body. So the gastrointestinal tract, the respiratory lining, the reproductive linings, these are places where viruses are likely to come into the body. And so we have the sort of extra system of interferon in those locations, namely type 3 interferon, interferon lambda, that gives us sort of extra antiviral potency in those locations, which is quite necessary and protects us from infection coming in through those anatomical sites. So we see that there are 3 subfamilies here, and from here we can dig in a little bit deeper to the kind of molecular signaling and how it's different and how it's the same across these families. This slide is a bit busy, but I think we can break it down piece by piece. One major point that I want to start out with is saying that interferons have a lot of overlapping signaling. They induce what I would call overlapping programs, primarily of JAK-STAT signaling. So this is something that interferons have in common with hematopoietins. Both families of cytokines use JAK-STAT signaling, but they can sort of specialize the outputs of that JAK-STAT signaling by using different submembers of the JAK and STAT families. So let's talk about what the interferons, how they use JAK-STAT signaling to induce antiviral responses. So laid out here are the receptors associated with the different families of interferon. For type 1 interferons, which is interferon alpha and beta, they activate a receptor known as IFNAR, or the interferon alpha receptor. And like many cytokine receptors, it exists in a dimer of 2 chains. One chain is known as IFNAR1, the other chain is known as IFNAR2. We typically just say IFNAR for short, and we sort of mean the complex of both 1 and 2 subchains. So this is the type 1 interferon receptor, and again, notice that it's associated with alpha and beta. In the middle here is actually the type 3 interferon receptor, the interferon lambda receptor. And its chains include one chain that we call the interferon lambda receptor, chain 1. Interferon lambda receptor is a bit unique though because it actually uses a receptor chain that it shares with many other cytokine receptors known as the IL-10 receptor 2 chain. This is a heterodimer of sort of 2 distinct subunits, but overall you can see that at least one of them is named interferon lambda receptor. That's because it is complementary to the interferon lambda cytokine ligand. Finally, the type 2 interferon receptor, which recognizes interferon gamma, it is also a heterodimer made up of interferon gamma receptor 1 and 2 chains. So you can see there's structural homology between all of these interferon receptors, and that structural homology is useful because that means that they can all interact with various Janus kinases, various JAK kinase molecules in order to facilitate the activation of STAT transcription factors. And one thing here that I'll draw your attention to is that both the type 1 interferon receptor and the type 3 interferon receptor both associate with 2 particular Janus kinases, JAK1 and TYK2. So TYK2 is also a Janus kinase, even though it doesn't have JAK in its name. You can see that in both cases, JAK1 primarily associates with STAT1. It primarily phosphorylates the transcription factor subunit STAT1, whereas TYK2 will phosphorylate the transcription factor subunit STAT2. And the result of the activation of IFNAR then is the formation of dimers or heterodimers of STAT1 and STAT2. This forms a functional transcriptional factor, a transcription factor complex, which can translocate into the nucleus and activate innate immune genes. And the types of genes that are induced by this response are those genes who have a particular sequence in their promoter known as the interferon-stimulated response element. So if a gene has an interferon-stimulated response element in its promoter, the STAT1-2 heterodimer can bind to it and drive its expression. And the set of genes that are responsive to this type of stimulation we refer to as interferon-stimulated genes, those genes whose expression is induced or stimulated by interferon signaling. So again, that is pretty similar between type 1 and type 3 interferon. There's a lot of redundant overlapping signaling going on here, but just recall that the main difference between these pathways is the localization of where these receptors are found in the body. IFNAR is found everywhere, whereas the interferon lambda receptor complex is only found in barrier tissues. We can contrast that with the type 2 interferon receptor, the interferon gamma receptor. It has a different set of Janus kinases, namely JAK1 and JAK2, and both JAK1 and JAK2 actually phosphorylate the STAT1 subunit of the STAT transcription factor complex. So the result of interferon gamma receptor activation then is the formation of homodimers of STAT1. See, that's in contrast to the STAT1-2 heterodimers that are induced by type 1 and type 3 interferon stimulation. So these STAT1 homodimers, they also translate into the nucleus, and they bind to a slightly different sequence in the promoters of various genes, that sequence being called the gamma interferon activation site, or GAS. And you can associate, again, gamma with type 2 interferon because that is interferon gamma. Genes that have GAS elements in their promoters are also referred to as ISGs. There's just going to be a different set of interferon-stimulated genes, although there's quite a bit of overlap between the genes induced by either the homodimers or the heterodimers of STAT1 and 2. So, summarizing some key points here, what I want to point out is that all interferons induce JAK-STAT signaling, and they induce sort of one of two major STAT transcription factor complexes, either homodimers or heterodimers. But in either case, the end result is going to be the very strong induction of genes that we refer to as interferon-stimulated genes. And these interferon-stimulated genes include very many very potent antiviral effector molecules, and they induce a state in the cell, as we'll see, which is very inhospitable to viral replication. And this is the main way that these pathways end up protecting the cell from viruses. So some additional points we can make about how this signaling operates and how it's regulated. A sort of somewhat unique, or maybe just a sort of special feature of type 1 interferon is that once we turn type 1 interferon on, typically it potentiates itself exponentially. There's a very strong feed-forward amplification of type 1 interferon signaling. So let's look at some of the molecular underpinnings of that feed-forward regulation. To induce interferons in the first place, usually we need to recognize non-self. Usually this is a non-self entity associated with a virus, some kind of viral PAMP, viral pathogen-associated molecular pattern, which is going to activate a pattern recognition receptor, again, that is sort of specialized for recognizing viral materials. But when that pattern recognition receptor is activated, that is going to set off a cascade to initiate the initial production of interferon. The way that looks is the activation of kinases like TBK1, tank binding kinase 1, whose main substrate, at least for right now, is a transcription factor known as IRF3, interferon regulatory factor 3. And you can see that when it is phosphorylated by TBK1, it also forms dimers, which translate into the nucleus. And there are many gene targets of IRF3, but a major one, for the purposes of this slide, is actually the cytokine interferon beta. So you can see that the interferon beta promoter, IRF3, is able to bind to it and activate it, which is going to drive the expression of that cytokine ligand, interferon beta, which will then be secreted by the cell. Interferon beta is then going to sort of float around, and if it interacts with its receptor, it can also transduce a signal. So in this example, we're sort of looking at autocrine signaling, meaning that the interferon beta is going to bind to the type 1 interferon receptor on the same cell. And when the type 1 interferon receptor is activated, as we saw on the last slide, that leads to the activation of JAK1 and TYK2, which leads to the formation of activated STAT1-2 heterodimers, which are going to translate into the nucleus and bind to the interferon-stimulated response elements in the promoters of various genes. One gene that has an interferon-stimulated response element in its promoter is another transcription factor called IRF7. This is an example of one transcription factor actually inducing the expression of another transcription factor. So IRF7, interferon regulatory factor 7, is then made and produced, and so then we find it in the cytoplasm after it's translated. And through some other mechanisms that I'm not going to go into just for the purposes of time and simplicity, IRF7 is then also activated, usually in the presence of interferon stimulation. And when it is activated, it goes back into the nucleus again, and it is able to induce the expression of the other type 1 interferon cytokine ligand, interferon alpha. Interferon alpha is then made, translated, and secreted by the cell. And remember that interferon alpha and beta have the same receptor, IFNAR. So interferon alpha can then go and activate IFNAR. And you can notice here that that leads to more STAT phosphorylation, which leads to more production of IRF7, which leads to more interferon alpha being made, which then leads to more stimulation of the type 1 interferon receptor. So you can notice that all these events are connected in a big loop, and that represents a positive feed-forward loop that is going to exponentially increase the amount of type 1 interferon that is being made and also transducing signals through the type 1 interferon receptor. So this is a very important way that we mount very fast and very sort of exponentially increasing antiviral responses upon some kind of viral infection. So we do all this pathway, we do it successfully, we make interferon in response to some kind of viral PAMPs. Who cares? What do these cytokines do that actually help us control a viral infection? Well, I mentioned that interferons ultimately are going to induce a class of genes known as interferon-stimulated genes. And as a set, interferon-stimulated genes represent effector molecules that induce something that we call the antiviral state, a kind of cell biological state in which various cellular processes are turned off or altered in a way that makes it very difficult for viruses to enter the cell or to reproduce themselves using the cellular machinery. So we can look at some examples of what the antiviral state is constituted by, as well as just a few examples of the ISGs, the particular interferon-stimulated genes that induce those various effector functions associated with the antiviral state. So the first one that we might mention is that interferon signaling and the antiviral state induced by it is associated with the blockade of endocytosis. So cells turn down their rates of endocytosis. And this is helpful because this is an effective way to block viral entry. Many viruses use endocytosis to gain access to the interior of the cell and infect it. So by turning down endocytosis, this is an effective antiviral strategy. And I should say that this is true not only in an autocrine fashion. So in our last slide, we saw sort of an example of autocrine interferon signaling, interferon sort of signaling back onto the same cell that made it. But interferon is also acting in a paracrine fashion and signaling onto cells in its environment that are uninfected, which have not yet seen any viral PAMPs. And this is sort of even more important because when we induce interferon signaling in uninfected cells in the environment and we induce the antiviral state in those uninfected cells, that's a very effective way of preventing viral spread. If any viruses are in the tissue, if we can turn down endocytosis in uninfected cells, that's going to be a very effective means of preventing new infections from occurring. However, assuming the virus has gotten into the cell and it is infected, there are still other things that we want to do. One is we usually turn on processes associated with RNA degradation and RNA editing. And a major effector molecule that accomplishes this are OAS proteins, which activate a molecule called RNase L. From the name RNase, you might be able to gather that it's a nuclease enzyme. It's going to sort of chew up RNA and degrade it. This is important because many viruses are made of RNA. They are RNA viruses, but even DNA viruses often have RNA intermediates that they use to transcribe and translate their genes and their proteins associated with their viral particles. And so by turning on processes by which we destroy RNA, typically we can prevent viruses from successfully replicating themselves genomically. And there are some processes by which these effector mechanisms that degrade RNA can be specialized to recognize viral RNA and degrade it at a higher level, then they might degrade host RNA. Although in general, host cells are they can sort of wait it out. They can sort of resist the state of having their RNAs degraded for a little while. Whereas viruses, that's kind of a catastrophic outcome to have their RNA being degraded. Okay. Beyond RNA degradation, we can sort of move down the central dogma just a little bit and think about also viruses need to translate their proteins, and they take advantage of host ribosomes in order to do so. So using an ISG known as protein kinase R, this facilitates an interaction that has the end result of turning down ribosome function, turning down translation. And so again, the cell can withstand this for a while, but when viruses are unable to translate the proteins that they need to successfully replicate and repackage themselves, that's a very effective way of kind of stopping them in their tracks and preventing the propagation of infection. Finally, we can also inhibit some of the last stages of the viral life cycle. So even if we successfully, or even if the virus successfully makes its proteins, those proteins need to be reassembled into a functional viral particle. And so there are special ISGs known as MX proteins, which also inhibit viral assembly. And so you can see we can target multiple stages of the viral life cycle: entry, transcription, translation, and assembly, using a complement of, or a diverse set of interferon-stimulated genes. And the activity of all of these things operating at the same time is sort of a biological state that we refer to as the antiviral state within the cell. So one last note about interferons is that all of these functions that I just described, they are pretty powerful, and for that reason, interferons have and are currently used as drugs, as disease-modifying agents, particularly against viral diseases as well as against cancer or neoplastic diseases. So after the discovery of interferon, it was very exciting because, as you can imagine, as it was learned that you're turning down transcription, turning down translation, that has clear antiviral benefits. But this also is potentially helpful in the context of cancer, because cancer is a state in which there is sort of too much cell division, too much transcription and translation happening. And so if you can target interferon to cancerous cells, that's also an effective means of turning down their ability to replicate and establish tumors and so on. And so interferons have been used historically in the clinic. You can see an example of an FDA-approved, or at least a previously FDA-approved interferon drug agent known as Interferon, which was a synthetic interferon alpha molecule. This is no longer widely used because many of the conditions that it was originally purposed for now have better and more specific therapies that we can use against them. And one of the reasons that we continue to develop better therapies for things that were previously treated by type 1 interferon is because, as I mentioned, type 1 interferon is expressed all over the body. And so even if you have a viral infection and you're treating with interferon, that can be antiviral and it can be helpful, but it also causes a lot of off-target inflammation and immune activation just because it's activating essentially cells in the entire body if you're taking a systemic interferon treatment. And so treatment with a type 1 interferon is usually not widely indicated now and is sort of only used when there are very few other options. However, I mentioned that type 3 interferons overlap with type 1 interferons quite a bit in terms of their molecular signaling. But when these interferons type 3 were discovered in the 2000s, as I mentioned, there was a lot of excitement because their expression is more restricted. As I said, they're only expressed in barrier tissues compared to the ubiquitous expression of type 1 interferons. That means that if you treat with type 3 interferons, you're sort of localizing that response only to those barrier surfaces, again, where viruses are very likely to be. And so you can avoid some of the off-target inflammation and side effect profile of treatment with ubiquitous type 1 interferons. And so while, as of the recording of this video, there are no FDA-approved type 3 interferon-based treatments, many of them are in clinical trials. And I pulled a recent example just from the literature, which you can see here for the treatment of chronic hepatitis D. And so this is a very promising sort of avenue in sort of harnessing elements of the immune system like interferons for the treatment of disease, sort of beyond their endogenous purpose in the innate immune response. Okay, let's summarize our discussion of interferons. I mentioned that they are important antiviral cytokines, and we saw that there were three types, 1, 2, and 3, which differ in their cellular expression. And generally, we can associate type 1 and type 3 with innate immune responses, whereas type 2 is more associated with adaptive immunity because of its association with T cells. Interferons collectively induce a robust antiviral and immunomodulatory transcriptional program, which is characterized by a set of effector molecules known as interferon-stimulated genes. Interferon signaling, type 1 interferon signaling in particular, is characterized by feed-forward amplification, and we sort of dissected some of the molecular mechanisms that underlie that. Finally, we said that the collective action of ISGs induces something we call the antiviral state, and this can happen in both an autocrine and a paracrine fashion, and it's characterized by a variety of processes, including, notably, a state of increased mRNA degradation and the inhibition of translation, which work together to prevent further viral replication. So that covers it for interferons. In our next video, we'll talk about yet another class of cytokines known as chemokines. See you in that video.